Hello, everyone, and welcome to Exam 1 for Anatomy and Physiology 2 review session. So, if you've had me before, this is just like my previous exam. So, starting with a quick overview here. There are 90 questions. 90 questions, and I grade it out of 85, so that means 5 bonus questions, mixture between multiple choice, true and false, and matching. And then don't forget, then there is the practical portion as well. So this exam, so we'll talk about each as we go through. This exam, remember, is from chapters 11 through 15. Chapter 11 is regions of the brain, types of neuron cells, and so forth. Chapter 12 is then the central nervous system. Chapter 13 is then the peripheral nervous system. Chapter 14 is the autonomic nervous system, and chapter 15 is special senses. This is one of the biggest exams in A&P, and this is probably going to be one of the harder ones. But I'm sure you all are going to do great. We're going to have a nice little review session right here on it all. So the first, stuff we talked about, what are the different neuron types? So that's going to be important. So you have bipolar neurons, you have unipolar neurons, and you have multipolar neurons. So know which neuron is typically involved in the special senses of sight and smell. We just saw that. Which one is found in that dorsal root ganglion? And then which one is the most common? So make sure you understand the definitions. There is a good virtual lab on that. So study tips for this exam. Look at the virtual labs again. Anything on those are, all those matching sections and so forth on those virtual labs. Definitions are all important to know. Quizzes, lecture quizzes, and also the Kahoots. I could pull questions from any of those things, so you have like your little question bank. In a way, that doesn't mean that's the only questions I can choose from, but that would give you a great start study-wise. Now, don't memorize answers. Make sure you're reading and trying to understand that question. So then we talked a little bit about action potentials, so make sure you understand the parts of an action potential. So if I were just to draw that graph out here real fast, and then we have time down here, we have millivolts over here. We start for a neuron at minus 70 millivolts. It's sitting there until we get a graded potential. It comes in, and once we hit minus 55 millivolts, that's when we hit threshold. So remember, that is the threshold right there. And then once it hits that, it's going to jump up to about plus 30 millivolts. And then it comes back down, drops down a little bit more, and then comes back up a little bit. It's not a good drawing there, but you know what I mean. So then time is in milliseconds down here as well. Now, the different parts of this. So remember, this is when depolarization happens. The depolarization is when sodium comes in. So sodium channels are open. And then here, sodium channels close, then calcium channels is open. So here then this is a repolarization. Now potassium now comes out. The potassium, as it's exiting, the charge drops back down. It drops all the way back down. It can go down to like minus 100. And then, so when it drops down really low, that's called, here, I'll draw this line a little different, like this, hyperpolarization. Oops. So make sure you understand the different parts of here. Then you have your two refractory periods as well. So you have the absolute refractory period. This is the point, this goes to about right here to when that action potential starts. This is when sodium channels no longer work. And then you have the relative refractory period, which is a time until it reaches back to that resting membrane potential again. So it can fire again in this region here, but it's going to take a strong stimulus for it to do so. So make sure you understand these different terms. It'll help you a lot and understand, polarization of a cell. If a positive ion moves in, what's going to happen? If a negative ion moves in, what's going to happen? So understanding how an action potential works threshold potential is going to be really important. And then remember with that, then you get to the synapse. So the synapse, that event, you get down there, just draw a horrible looking synapse here. So action potential is coming down through here. So here's, you have sodium and potassium coming in and out. So sodium is rushing in, potassium is coming out. But then down here at this synapse, let's draw the next cell down here. Remember, you guys did draw a synapse too. Now remember, the cell doesn't end there. I'm just drawing that line there for simplicity. At the end of the synapse, the ion channels change. When that action potential reaches down here, calcium 2+ then rushes in. So make sure you know that calcium rushes in down here, which then causes these neurotransmitter vesicles to fuse and release neurotransmitter. Neurotransmitters are things like acetylcholine is one of the big ones, ACH, that binds to then receptors on the other side, and those receptors could be excitatory or inhibitory. If they are excitatory, they're known as an EPSP, and that means they're causing a depolarization in this next neuron, or they can be an IPSP, which is an inhibitory, which means they are polarizing. So if you just look at polarizing here, that's what an IPSP would do. So preventing an action potential. So make sure you understand the steps in synaptic transmission there and how some neurotransmitters could be excitatory and some could be inhibitory. It all depends on this receptor right there. Now, I'm not going to ask about specific receptors and say, is this one excitatory, is this one inhibitory? Just know that it does depend on that receptor. And then also the strength of a stimuli. So what makes a stimulus stronger or weaker? So remember, it's the frequency of action potentials, not the amplitude. This amplitude of an action potential is always the same. It's how many, so, is it like this? You draw a wave, or is the action potential really soon, right after the next one, after we can get to that next period? So it's the frequency in which they can reach the destination. That is what determines how strong a stimuli is. Now, what else do we have with that one? So the different cell types, remember, or the different terms for the different cells. So a cluster of neurons in the peripheral nervous system, remember, is called a ganglion. In the central nervous system, it's called a nucleus. Or, like the suprachiasmatic nucleus is a cluster of ganglions. So just terminology-wise, make sure you understand those differences. And then understand the structure of the route into the spinal cord. So here, I don't want to draw a spinal cord, but remember, you have that nerve coming in. You have that one that has a bulge. That's the dorsal root ganglion. It comes into the spinal cord, and then you have that area of white matter on the outside. Let me draw a horrible spinal cord here. And here is the little central canal. And then here is, oh, look, we can draw gray matter. Let's then draw gray matter in here. So here there's a horn here, a horn there, and a horn down here. And then this gray matter comes through, another horn on this side, another horn here, another horn up here, and then another little canal coming in. So there's our little area of gray matter. Here, I'll even color it all gray for you. And then remember, it's white matter outside of here. So if let's go back to the white pen. If we're, a signal coming in, then it's going to synapse here in this gray matter. And remember, this is the dorsal horn. Then remember, this is the DRG, or the dorsal root ganglion. And this is the afferent division. So this is sensory input. This is all sensory coming in. This could be somatic sensory or visceral sensory. Then it, synapses here, goes through an interneuron here. And then, depending on whether it is autonomic, if it's autonomic, it's going to go out the ventral horn. So the ventral horn is going to synapse here and then exit. So that's for autonomic, so typically visceral sort of autonomic reflexes. Then if it's a motor output, it's going to synapse down through, so this one's the lateral horn, and then this one is the ventral horn. So ventral horn then synapses here and then exits. So then they combine back up and then come back out. So let's draw this as a nerve cord there. And then they combine, and then at the point they combine, that's when it's referred to as a spinal nerve. So there's central canal. This connection between the two, remember, is called the gray commissure. Commissure. So just important parts of a spinal cord. That is a beautiful spinal cord. I do say so myself. Very, very perfect. Remember, out here then we have the white matter. That's where you have the ascending and descending tracks as well. So that's important. You have the different cells in the actual brain and the central nervous system as well. I know I jumped around a little bit here going into the spinal cord. But back to the brain then, you have your different glial cells. You have the astrocytes. You have the oligodendrocytes. You have your other ones. Your ependymal cells. Ependymal cells. And then your last one are your microglia. So microglia, these are your immune ones. These are the ones that are moving, the ciliated ones that are moving in cerebral spinal fluid. And then these ones are the myelin forming cells of the central nervous system. Then the astrocytes are the ones there forming the blood-brain barrier and holding everything in place. So they're supporting cells. So make sure you understand the different cells. Remember, oligodendrocytes are similar to Schwann cells, but Schwann cells are only found in the peripheral nervous system. They are not found in the central nervous system. Oligodendrocytes are only in the CNS. Schwann cells wrap individual sections of an axon. Oligodendrocytes can wrap multiple axons in myelin. All right, so what else here? Oh, the different regions. Remember the so the space between the dura mater and the vertebrae. So back here, then you'd have the dura mater out here, and then the vertebrae out here. Oops, I switch colors here, and then in here, then you have that layer of fat and blood vessels. I'm not good at drawing this, but remember that layer of fat and blood vessels is called the epidural space. Epidural space. That's an important one. Then as the spinal cord goes down, remember that section of nerve fibers that extend below the conus medullaris, which is the end of the spinal cord, is then called the cauda equina. It literally means horse's tail because that's what the spinal cord looks like in that region. Kind of neat how it gets that name. So that's that. And then just some differences here. So just make sure you understand the key differences between the central nervous system and the peripheral nervous system. So like the sensory portion of the peripheral nervous system carries electrical signals toward the central nervous system, and then the motor portion carries electrical signals away from the central nervous system. So just like I was talking about earlier, and important differences there. Then, of course, you have the brain anatomy and brain region. So like what's on the brain stem. So that's the midbrain pons and medulla oblongata. Know the locations of those. Now, I'm not going to get super specific on the practical, and so like all the different association centers and regions like that. It's the main lobes I'm focused on. So like parietal lobes, frontal lobes, occipital lobes, cerebellum, midbrain pons, medulla oblongata, thalamus, hypothalamus, the main ones, corpus callosum. Remember, corpus callosum is what connects the left and right hemispheres of the brain, does not connect the cerebrum to the cerebellum, which sometimes make that mistake and think that, but its left and right hemispheres are connected by the corpus callosum. So that's the big one for brain anatomy and spinal cord. So know the structure of the neuron. Know the different parts of a neuron. I'm not going to draw a neuron here. I want to be too good at it. Let's try it. So here we have a neuron. These are the dendrites coming into this neuron here. Draw another dendrite down here, and then one more long one down here. Then we draw this neuron, and it all comes down through here to the axon. And then it can branch off to multiple synapses then. I'll just draw multiple little synapses down here, and boom. So this is a general structure of a neuron. So right here we have the cell body. So remember, this is cell body. I'm not going to label it all. I'm just going to do a quick little discussion on it here. Cell body has, nucleus in it and so forth. These are all the dendrites. And then around each of these, we have the little wraps of myelin. Let's see how good I can draw these. This one's looking good. Remember, this myelin is that highly layered, and we have so there is one Schwann cell, and then inside then is that myelin protein. So myelin lipid protein that wraps in there. This is then the axon. Oops, hit my mouse. This is the synapse, or multiple synapses. So, you can have up to 1,000 different synapses. This is the axon hillock. And since I labeled this much, I might as well just write dendrite up here. And so, those are the main parts of a neuron. Then inside here, there might be a little mitochondria moving around. There are these little nissl bodies you might see too. But here, like mitochondria, synapse, axon, Schwann cells, axon, hillock, cell body, dendrites. Those are the important ones. You should know all those words there that I just said. So sorry if you hear a dog barking. I think the mailman is coming by. Or a roommate is home. Yeah, good background noise. All right, and then we get into, so a lot of that you might see on the practical portion too. And then know a little bit about what some of the regions do and their importance. So the hypothalamus is a pretty important region. It's involved in many functions, including temperature regulation, water balance, and fat and carbohydrate metabolism too. Is when we get to the endocrine system, we're going to learn about how it plays a very, very important role in hormone production as well, and hormone release. And then the cranial nerves. Yes, we do need to know the cranial nerves and what they do. There are 3 cranial nerves 3, 4, and 6 that go to the eye. Remember the trochlear, abducens, and ocular motor. So there's something like that. And then back to the brain, I forgot to say this. So the hypothalamus has this little stalk that connects to the pituitary gland. It's called the infundibulum. Here, I'll write that. Infundibulum. Fun little word. And that one's going to come back again later in the endocrine system. I forgot to mention that one right now. But back to the nerves. Remember, you have your cranial nerves. So remember, we have 12 different cranial nerves. Now, you don't have to remember which are sensory, which are motor, and so forth. Just be familiar with them. And I'm not going to label any particular ones on the practical, so you can thank me later for that. But understand, just the basics of them. And then you have the different nerve plexuses, like the lumbar plexus, sacral plexus, brachial plexus, cervical plexus. Know, a little bit about each one of those, and what affecting each one could cause. So striking the funny bone or the ulnar nerve may cause an injury to what plexus, which would be the brachial plexus, and so forth. Or the phrenic nerve. Remember, the phrenic nerve is any spasms in that lead to hiccups because it goes to the diaphragm. So, little things like that about each of the nerve. You don't have to remember, how many branches they have, or they go from 5 to 3 to 2 and the different things like that. No, I'm not getting that specific on it. I taught that because you need to know it. They do that. Then remember, so with the senses then, you have the olfactory system, you have hearing and balance. So those are all cranial nerves too. Vestibular cochlear, olfactory, hypoglossal goes to the tongue. Now the vagus nerve, remember the vagus nerve is the only one that then goes below the neck region. Very important one in the parasympathetic nervous system. Let me check something. Oh, then we have the different pain receptors there as well. Remember, nociceptor is pain, thermoceptor is temperature. I know I stopped writing here. So different receptors. So remember, receptors are based on, type, location, and so forth. So interoreceptors versus exteroreceptors. They could be encapsulated or not encapsulated. So like nociceptors for pain reception are not encapsulated for recepting pain, whereas pressure receptors like the Pacinian or Meissner's corpuscles, they are encapsulated. Now you don't have to know the specific ones, but know the general overview of the types and the locations. Photoreceptors, those are what are found in the eyes. Chemoreceptors are found for chemical receptors. Little things like that might pop back up here and there throughout the exam. And then, so when you strike your funny bone, remember you're hitting your ulnar nerve, the one that causes impingement during carpal tunnel. That's your median nerve. So like we talked about some of the nerves and their importances. So little things like that might come back up as well. Now, one big thing with the peripheral nervous system we talked about too is that it had some regenerative capability. And remember, that is because of Schwann cells. So Schwann cells are what provide regeneration to a limited amount in peripheral nervous system. Oligodendrocytes do not do regeneration in the central nervous system, unfortunately. And then make sure you understand this little reflex arc right here. So it's the afferent division, goes through the little integration center here. Out here, there was a sensory receptor, little integration center. Then we get a motor output, which goes out the efferent neuron. And then we get the actual effector. Effector is then the effector organ, muscle, gland, etc. So make sure you understand that terminology when we talked about that reflex arc. You might see it again. And then, of course, then we get to autonomic. So know the differences between parasympathetic and the sympathetic nervous systems. So there are some matching sections on that. Which one increases blood pressure? Which one's maintenance? Decreases heart rate? And so forth. And then the other differences between sympathetic and parasympathetic anatomically. So which one has longer preganglionic fibers? Which one has longer postganglionic fibers? And then, there is that asterisk. So remember, all preganglionic fibers secrete acetylcholine. And then most postganglionic fibers in the sympathetic, all postganglionic fibers in the sympathetic pathway secrete norepinephrine, except for one. Sweat glands secrete acetylcholine. So I dwelled on that a lot in class, so it's pretty obvious it's going to come back again. So sweat glands equal ACH, or acetylcholine. And remember everything that's controlled by autonomic nervous system. So glands, smooth muscle, cardiac muscle, but not skeletal muscle. Skeletal muscle is voluntary, so we control that. Remember that like I was saying, that vagus nerve is the major route for parasympathetic outflow from the head, because it's the only cranial nerve that goes below, and it can get those signals out there faster for going out. And remember, it's parasympathetic because it's above and below the sympathetic. So we have to have nerves that come from above. The one that comes from above is that vagus nerve. And then there are the different fiber types, so make sure you understand what adrenergic is. So remember, adrenergic means they release norepinephrine or epinephrine. Cholinergic. So then we have a cholinergic. Oops, I need to spell this right and at least write it nice. Cholinergic then releases acetylcholine. So the preganglionic axons, the sympathetic and parasympathetic, they are cholinergic fibers that release ACH. And the sweat gland one is then also a cholinergic fiber. If it releases acetylcholine as a neurotransmitter, it's cholinergic. If it releases norepinephrine and epinephrine, it's adrenergic. And then remember, for pupil size, sympathetic one, think of a cat, sympathetic fight or flight. Cat's eyes get super dark and wide, so that is pupil dilation. So remember, sympathetic causes pupil dilation, parasympathetic causes pupil constriction. So when you're trying to focus for close vision, that would be a parasympathetic response, whereas really wide pupils for fight or flight is then sympathetic response. And then remember what the boss of the autonomic nervous system is. We all should know this one, the hypothalamus. The boss of the ANS. That's the main control center. So it can override inputs, can control outputs, and can add associations to things. So you see someone's making a frightening face, you then get frightened without having that fear because of the association of seeing that frightened face. You understand, oh, I should be scared. So fight or flight then kicks in the sympathetic nervous system, and then you're ready to go. And that's all directed towards the hypothalamus, interacting with limbic system and so forth. Really neat how that works. And then we are about to get into sympathetic, parasympathetic. Remember, sympathetic nervous system, most of its ganglia are in the sympathetic trunk or those collateral ganglia, whereas the parasympathetic has its ganglia closer to the organ it innervates. Then we get finally to these special senses, which we just recently talked about. So just different sections in here. Understand different parts of the eye. What's the sensory layer, which is responsible for focusing light rays on the eye, or like the lens. Then you have the aqueous humor, the vitreous humor, that fovea centralis around. So the fovea centralis, where the greatest visual acuity exists, right next to that macula densa there. So there, understand that is in the sensory layer of the eye being the retina. Now remember, so then the eye you have both cones and rods. Cones, we have 3 of them for color. Rods are then dark light. So then which are light sensitive and light insensitive, or which ones adapt quicker. Cones adapt quicker to light. Rods don't adapt quicker. They're actually bleached in light and take longer to adapt. Now I know we talked about a really complicated pathway for how rods work. You don't have to know that. Just know 3 cone colors, and why we can see several thousand shades of color. It's because this perception is perceived by various combinations between the 3 cone types. So if we draw the visible spectrum here and the 3 cone types, it looks something like this. And if you're looking at a color right here, this one's going to be about that, strength and activation. This one's going to be there, and that one's going to be there, and that's perceived by whatever this color is. Maybe it's yellow or orange. Remember, you have red, green, and blue cones. And same thing with smell in a way too. So smell is very unique because one single distinct smell is a combination of a variety of chemicals. So it stimulates different combinations of olfactory receptors all at once. So you're able to perceive that as that unique smell. And then, so back to vision, when you shine light into the eye, both pupils will constrict. Ever notice that? I didn't mention this one in class. I talked about shining a light in the eye, but I forgot to mention that both pupils will actually react. And that's because of that optic chiasm. So sensory input from the retinas of both eye actually converge at that optic chiasm, and the information from each eye is then delivered to both and left sides of the eye. So that's kind of cool. So remember, for vision, if these are the 2 eyes, back here, this optic nerve comes back from each eye here, and then crisscrosses at that optic chiasma. And then there's some nuclei right off to the side here that actually can summarize on those nuclei and send that information back out to the other eye or the other eye. so they can react through those ocular motor nerves and so forth. And then as they cross through, one crosses over to here, and then one actually goes to this side depending on the field of view they're looking at. And then over here, this one will cross over to this side, and then this one will cross over to this side. Then they'll summate on the back of the occipital lobe here. There's also another nuclei in here called the LGN, or lateral geniculate nucleus, but you don't have to know that. Oh yeah, these are eyes here too. You didn't know. Those are really good eyes. And then right here, that optic chiasm. Oop, there's an S in there. That's that crisscrossing, but that allows both eyes to react the same. That's why your eyes work in unison. If your eyes don't work in unison, that can create cross-eyed or double vision. Then for taste, you have the different taste sensations: sweet, umami, sour, salty, bitter. So understand the chemicals that cause each. So like sours, acids. Umami, that's monosodium glutamate, or that glutamate taste. Bitter, alkaloid. Salty are metal ions. Sweet, so like sugar and some lead salts. I understand it all depends on different receptors it binds to, and that's what activates those receptors. And then for hearing, remember, receptors for hearing are in the cochlea. Receptors for balance are then in the semicircular canals. And then in your vestibule, then you have your ear stones. Remember, those are made up of otoliths. So those little ear stones, little calcium carbonate crystals on that membranous bed embedded in the little cilia structures. When that moves left or right, it sends information about the position of your head in space. So these are called otoliths. Or your ear stones. And then that tube that connects the middle ear with your pharynx, remember that's the pharyngeotympanic tube, eustachian tube, or auditory tube. Any of those will work. The canal that comes in is the external acoustic meatus. That vestibule then is the one that contains the utricle and the saccule, which are these little ear stones here. And then, so that vestibule also is what detects linear acceleration. So very important. And then the main membrane as you come into the ear is the tympanic membrane. So here, we have this beautiful ear here. Here's the external acoustic meatus coming in. So it travels through there. The sound vibrates off this tympanic membrane right here. Tympanic membrane is connected to the hammer, which is connected to the anvil, which gets connected to the stirrup. So this is like a little stirrup here. This is the inner ear, the middle ear right here. This is the outer external ear. And then this is the inner ear over here, where it then goes into the vestibule with the semicircular canals. And then right here would be the cochlea, which is the little spiral structure. And then comes out then the little round window. This is the oval window. This is the round window. And then this is then where the eustachian tube then drains out down below. You look, I could draw in class. This works great. But yeah, these are little middle ear bones here that control and help make it so you don't damage this tympanic membrane. And then what else we have here? Remember that biological clock? We talked about it a few times. That's the suprachiasmatic nucleus. And then, one receptor I didn't mention earlier, mechanoreceptors. So these little cilia here are actually mechanoreceptors. If we expand one of these cilia really high, so if this cilia moves, it's attached to the cell membrane. The movement of this cilia actually opens channels that bring something like sodium into the cell. So these are, that's a weird M, mechanoreceptors. I never really got into detail of that, but that movement of the cilia actually opens or closes these ion channels, which then causes depolarization. Depolarization, I can spell. Perfect. And then think about what would happen if you lose hearing in just one ear. What would be the result of that? So remember, for localization of sound, your brain is able to analyze the difference between the left and right ears, and that short time difference can be figured out and picked up. So if you lose hearing in one ear, it's difficult to localize sounds. And same thing, if you lose vision in one eye, it might be difficult to have depth perception. Yeah, you can get used to it, but it is difficult. But yeah, that's most of this. Where is sound generally perceived? Remember, that's temporal lobe. Vision is occipital lobe. So things like that are important. Taste is in the insula. For the special senses, I think those are the main ones. Remember the difference for the retina, which one's the inward-facing side of the retina and the outward-facing side of the retina. So the outward-facing side of the retina are where the photoreceptors are. So if you were to draw the retina, the photoreceptors, so this is the back of the retina, and this is the layer right here, the photoreceptors are actually on the back side. And then, remember, they connect to those unipolar neurons and then connect to the ganglion cells, and then they travel out and out the optic nerve. So they all travel out like this and then combine down. So if I keep drawing these here, and draw more down here, this is what the back of the retina looks like. They all converge down the optic nerve. So this is then the blind spot right here, and light actually has to go through that to the photoreceptor, and then it goes back out this way and back out. So it's a little backwards there, and then this is also why we have the blind spot. And then there's that pigmented layer of melanin back here to help absorb the light so it doesn't activate other cones or rods. But I think that's a good start. Understand the main models, so like the neuron model, the spinal cord model, the brain model, the ear model. Yeah, those are the big ones. And there was the eye model, which, yeah, there might be stuff on that one too. So those are the main models to study, and all your virtual labs will help with that. But overall, this is all I have to talk about for this little review session here, and I hope it helps you. I know I just gave you a lot that you need to look into. It wasn't all-inclusive here, so make sure you review your lecture quizzes, your virtual labs, and those Kahoots. All will help you tremendously on this exam. If you have any questions, feel free to let me know. But that is all I have for now. I hope you have a great day. Good luck studying, and bye-bye.